Hello to all the viewers. Today I have the pleasure to talk to Professor Daniel Otzen from iNano at Aarhus University. He will tell us more about the usage of FIDA technology for studying intermolecular protein interactions. Daniel, would you please tell us more about yourself and your laboratory?
Yes, Maya, I'm very happy to do so. So I work with proteins and how they affect good things and bad things in life, you might say. I've got two main areas of research. One of them is to understand protein aggregation in diseases like Parkinson's disease and what you can do to avoid that, or at least to somehow suppress it. And the other one is to harness the power of enzymes to do all sorts of useful things for society. Enzymes that work at low temperatures and increasingly enzymes that are able to degrade plastic. So they're quite different areas of research, but FIDA is useful for both.
Wonderful. And what questions and challenges are currently being addressed in your field of research, and how can FIDA address these?
So in Parkinson's disease, we're interested in understanding what makes these aggregates harmful. Aggregates can be insoluble when they accumulate in the cell, and then they're just large and lie around and don't do much. But they can also be small, soluble complexes that we call oligomers. They can move around quite quickly and interact with a lot of things. And in that case, FIDA is really, really good because the oligomers can be made in the lab, but you can't make a lot of them. So you need to have a technique that can work with small concentrations and small volumes. And FIDA is second to none there. And you also want to work with it under conditions where you don't manipulate these oligomers. You don't bind them to a surface or immobilize them in different ways. And again, FIDA is really great here because it allows you to have it in solution and then expose it to other components. And thirdly, it has to be under truly equilibrium conditions. So the oligomer is exposed to an appropriate sea of potential binding partners. And all this is something that FIDA provides.
Wonderful. So what, if any, capacities were you missing in your field of research, and how did FIDA address them?
Yeah, so particularly for the oligomer interactions that I mentioned, FIDA has really been a game changer, I think, because, as I mentioned, it allows you to measure correct or robust equilibrium affinity constants for how oligomers can bind to whatever you expose it to. But it also allows you to understand the basis for how oligomers work. For example, we know that they can bind to membranes and lead to disruption of them so that things can leak out. But there is a difference between binding and leakage. And by having the FIDA technique, you can actually directly measure if the oligomer is binding to the membrane or not. And that is really useful. It's all in solution, but you still get this readout there. So that's extremely important. And also for enzymes, they can occur as monomers, as dimers, as tetramers with different levels of activity. And again, FIDA is really great because, of course, it provides information about the size, and you can really go down in extremely low concentrations to find out how the concentration affects the association of the protein, and then you can build up models of how they look like.
Interesting. And in light of other technologies that you have in use at the moment, how does FIDA fit into the challenges at hand?
Yeah, so what's really also great about FIDA is its specificity. And you just need to have a fluorescently labeled protein of interest, such as the oligomer or an enzyme. And then you can expose it to a very complex mixture and see whether it binds to something in that mixture or interacts with it or not. And that mixture will be invisible in FIDA because the components are not fluorescently labeled. And we've actually done that to show, for example, how the oligomer can bind to different components in a cell lysate, where there can be thousands of different components. And we get information like which of course does not allow us to identify the binding partner, but we can then go in and manipulate that with, say, components that should perhaps bind to the oligomer and prevent it from binding to other components. So it simply provides readouts under realistic physiological conditions, I would say.
Wonderful. And how does specific data that you obtain through FIDA contribute to the studies that you have earlier mentioned?
So what is also great about FIDA is that it allows you to basically run a lot of samples through at the same time. It's got an autosampler, of course. And we've recently completed a study where we looked at more than 30 different monoclonal antibodies and how they interact with the oligomer. And we could measure all of their interactions with the oligomer at no particular cost of either antibodies or oligomer, simply because FIDA is such a economical technique for measuring these things, and we can also compare them rigorously with each other under exactly the same conditions. So that has also really made a difference for us in our understanding and our ranking of these different antibodies. And I believe that FIDA could really become a basically standard technique within the pharmaceutical industry to assay interactions between biologics and their targets, and also check, for example, how this is affected under different conditions, but also to compare different components to get baseline values here.
Wonderful. Thank you so much. And my next question is a bit funny. What would you miss the most if FIDA was to disappear from your lab? Considering you mentioned so many different things, I wonder what's the most important.
Yeah, as you can see, FIDA comes as a package with many different advantages. I think that what I really like about FIDA is that you get a very understandable readout. You get the hydrodynamic radius. Many techniques only provide an indirect readout. So FIDA's very straightforward and I think understandable and also testable outcome of the hydrodynamic radius is so important. And then added to that, the fact that it's under equilibrium conditions at little cost, and you've got high throughput or at least medium throughput options, they all help out. But I think that the fact that you get a truly molecular picture of what's going on is unique for FIDA.
Wonderful. And would you mind telling us what plans do you have for applying FIDA technology moving forward?
Yeah, so that's a great thing, because the challenge is just to ensure that whatever you want to measure can fit into the capillary and gives a decent signal. And I'm increasingly working with enzymes that break down plastic. The interesting thing is that this is a pretty slow process because enzymes are not designed to break down plastics, but they can to some extent. So there will be rather stable complexes between enzymes and plastics. Plastic is insoluble, obviously, but we can before we expose the plastic to enzymes for analysis, we can break them down to manageable, suspendable sizes. And then I'm really keen to see how we can monitor the depolymerization or the sort of increased size reduction of these small plastic pieces by FIDA in a very direct fashion. So that's my next ambitious step.
It sounds very impactful, and we hope it works well, because it sounds like a wonderful piece of research and everything it can bring afterwards for the environment. So that's fantastic. And thank you so much for all.
Actually, there is one more thing. I'm sorry for butting in, but this is just
that's perfect.
Sound of my enthusiasm. I also know that you have a new kinetics package to work with, and I also think this will just be adding to the really useful properties of FIDA, because so far it's all been equilibrium. But if you have, say, slow dissociation or association steps in whatever context, I'm really looking forward to trying that out too. So that was an extra footnote.
Fantastic. Thank you so much for all the insights and the interview. I hope that our viewers find it useful, and I'm pretty sure they will. Thank you.
Thank you. Pleasure.